Hello, welcome to this Frontiers in Science deep dive session on precision cardiovascular medicine. I'm Lee Baker, the Editorial Advisor at Frontiers in Science. It's fantastic to see so many people from around the world on this session today. Welcome, everybody. If you're new to Frontiers in Science, this is our flagship multidisciplinary open access journal at Frontiers. We publish exceptional peer-reviewed lead articles that are addressing transformational science key to global challenges in human and planetary health. We publish these articles within unique hubs of multi-audience content designed to increase their reach and impact across scientific audiences together with policy audiences and broader society. Today's deep dive focuses on this lead article, Precision Cardiovascular Medicine, Shifting the Innovation Paradigm. Cardiovascular disease remains the leading cause of death worldwide, despite decades of progress in diagnosis and therapy. Conventional one-size-fits-all therapeutic approaches still leave many patients at risk. Meanwhile, advances in systems biology and network science, enabled by multi-omics, bioinformatics, and artificial intelligence, are now revealing the complex heterogeneity that underlie cardiovascular diseases, and they're opening up a new era of precision medicine for cardiovascular medicine. In their lead article, Professor Masanori Aikawa and colleagues explore the scientific and technological advances that are enabling this transformation and the prospects for its implementation throughout the research and innovation process. Here is the lead article in its article hub. Alongside the lead article, you have two editorials, one by Professors Valentin Fuster, Philip Sawersky, and Girish Nadkarni from the Mount Sinai Health System, and another by Professor Joseph Wu and Dr. Si-Yon Ri from the Stanford University School of Medicine. There are viewpoint articles by peer reviewers Professor Hugo Ten Cate from Maastricht and Professor Rainer Schulz from the Justus Liebig University Gießen in Germany, and a policy outlook by Professor Amam Mbakwem from the University of Lagos, Vice President of the World Heart Federation. Many of these contributors are with us today. As always, the hub also has a lay explorer video and an article in Frontiers for Young Minds, our science journal for kids. So this is today's agenda. First, we're going to hear from four authors of the lead article, Professors Masanori Aikawa, Joseph Loscalzo, Chiara Giannarelli, and Jean-Luc Balligand. Then we'll have a panel discussion featuring also Hugo Ten Cate and Professor Amam Mbakwem. So first up, I'm delighted to invite the lead author of the article we're going to talk about, Professor Masanori Aikawa. Mazar is Professor of Medicine at Harvard Medical School and the Yoshishiro Miwa Endowed Chair and Founding Director of the Center for Interdisciplinary Cardiovascular Science at Brigham and Women's Hospital in Boston. His systems-based cardiovascular research integrates multi-omics, network science, single-cell analyses, and machine learning to identify new therapeutic targets. He's won various awards from the American Heart Association. He served as president of the North American Vascular Biology Organization, and he's the field chief editor of Frontiers in Cardiovascular Medicine. Masanori, thank you for bringing this topic to Frontiers in Science. The floor is yours.
Thank you, Lee, for kind introduction. Cardiovascular disease remains a major global burden and leading cause of death, accounting for 19 million deaths worldwide in 2020. This number is projected to rise to 26 million by 2030, despite enormous efforts that medical and scientific communities have taken that led to substantial progress in understanding disease mechanisms and risk factors, and effective therapies such as statins. Significant challenges persist. These include residual cardiovascular risks, heterogeneity of disease mechanisms, several challenges in drug development, and insufficient cross-disciplinary and cross-sector collaboration. But we see these challenges as opportunities that drive the development of new solutions. Despite population-based risk-modifying drugs such as statins, substantial residual cardiovascular risks persist. This highlights the need for precision medicine as an additional layer of protection beyond one-size-fits-all approaches. Growing evidence also shows that inflammation plays a central role in these remaining risks. Thus, my group has focused on the biology of macrophage activation in the last couple of decades. No matter where they come from, macrophages play an important role in the pathogenesis of multiple vascular diseases, including atherosclerosis. Accumulating evidence suggests macrophages is a heterogeneous population. A traditional theory proposed two macrophage subpopulations, pro-inflammatory M1 and anti-inflammatory M2. However, more recent evidence shows that macrophage heterogeneity is far more complex than M1-M2 dichotomy and involves multiple subpopulations. Our current theory is that when a pro-inflammatory subset predominates, the plaque microenvironment becomes pro-atherogenic, whereas the dominance of an anti-inflammatory subset creates an atheroprotective environment. Due to multiple factors, including genomic characteristics, exposome characteristics, and multiple risk factors and pathologies. And human cardiovascular diseases are extremely heterogeneous at molecular, cellular, and patient levels. To tackle this challenge, a systems approach involving multi-omics, single-cell analysis, and network medicine helps to further understand cell heterogeneity. Clinical information analyzed by AI would also contribute to patient stratification. Biomarkers that represent a target subpopulation helps to translate the biology of heterogeneity in subpopulation-targeted precision medicine. I'm going to provide a couple of specific strategies. The single-cell analysis can help us identify the disease driver cell subpopulation. In parallel, analyzing the clinical data and information using AI may help us identify the high-risk patient subpopulation. Then, such an information of human heterogeneity would help us develop subpopulation-targeted therapies. Another strategy might be using the information of macro cell heterogeneity into drug discovery. In this publication in Circulation 2023, we use single-cell RNA sequencing data in computational drug screening to try to identify compounds that can reduce the expression of genes in pro-inflammatory macrophage subset and increase the expression of genes in anti-inflammatory macrophage subset. We identified multiple compound, including this BI-2136. In vitro validation showed us that this compound can actually shrink pro-inflammatory macrophage subset and even expand the anti-inflammatory macrophage subset. We furthermore conducted multiple in vivo experiments to show that this compound indeed can shift the balance between pro versus anti-macrophage subpopulations and enhance the anti-inflammatory or atheroprotective macrophage functions, including efferocytosis, and ultimately retarded the development of atherosclerotic plaques in mouse models. Another challenge is insufficient collaboration between disciplines and sectors. There's a huge gap between academia and industry in the conventional model of drug development due to insufficient expertise and resources in academia and unwillingness to take risks on early projects in industry. So many promising targets that are identified in academia cannot go through this valley of death. We therefore believe that a new model by which you can fully integrate strength of academia industry in drug development is needed. So we therefore launched the Center for Interdisciplinary Cardiovascular Sciences, or CICS, in Brigham Women's Hospital and Harvard Medical School in a collaboration with Japanese pharmaceutical company Kowa in 2009 as a new model of academia-industry collaboration in drug discovery. Another key solution to facilitate cardiovascular innovation is integrating data science into the biology. At CISS, our research represents a dynamic interface between cardiovascular biology and data science. Each project typically starts with omics screening, followed by target prioritization by bioinformatics and prediction of target disease association by network analysis to try to increase the success rates of new targets in future clinical stages. On this slide, I like to emphasize one thing. A systems approach does not replace a traditional approach. For example, in our case, after we identify the potentially promising targets, we conduct comprehensive in vitro and in vivo validation studies using conventional method. In addition, accelerating precision medicine will require multidisciplinary and multi-sector partnerships on a global scale, including intergovernmental coordination. The COVID-19 pandemic demonstrated how rapidly new therapies can be developed when diverse sectors unite toward a shared goal. Open science policies and data sharing were also central to that success. Although COVID-19 demanded global cooperation due to its acute and immediate impact, cardiovascular disease is a major global burden that warrants large-scale collaboration. Importantly, despite carrying a disproportionate share of cardiovascular diseases, the global south is often left behind. In their viewpoint related to our article, Dr. Sion Lee and Joe Wu emphasized that perhaps the greatest challenge ahead is ensuring global equity in access to precision therapeutics. They also noted that while traditional drug development and approval processes assume patient homogeneity, precision medicine requires new regulatory frameworks capable of evaluating targeted therapies for patient subgroups, along with global leadership and network to make this possible. To conclude my talk, I'd like to briefly discuss future perspectives. I borrowed this figure from Dr. Rainer Schultz's viewpoint related to this our article. To successfully advance precision cardiovascular medicine, more clinically relevant experimental models will likely be required. Recent target discovery research has increasingly focused on cell type specific biology, highlighting the need for developing cell type specific drug delivery systems. New drug modalities such as RNA therapeutics continue to emerge, integrating technologies like AI to analyze complex regulatory networks and to more accurately predict potential off-target effects may thus become essential. Again, we strongly believe that seamless collaboration across disciplines, sectors, and countries will create an effective synergy between emerging technologies and new paradigms, accelerating the translation of discoveries into precision cardiovascular medicine. Thank you.
Thank you very much, Masanori, for that beautiful introduction. I'm sure we're going to touch on many of these topics later on in the panel. For the moment, our next speaker is Dr. Joseph Loscalzo, Samuel A. Levine Professor of Medicine and Hershey Distinguished Professor of the Theory and Practice of Medicine at Harvard Medical School. He was also former Physician-in-Chief and Chair of the Department of Medicine at Brigham and Women's Hospital in Boston. Joe is a renowned cardiovascular scientist, clinician, and teacher with numerous awards, high-profile appointments, and influential publications over a long and distinguished career. He's particularly known as a founding figure in establishing the field of network medicine, together with pioneering work in vascular biology, redox biology, systems pathobiology, and microRNAs recently. So, Joe, welcome to Frontiers in Science. Welcome, and over to you.
Thank you very much for that kind introduction, Lee. I'm delighted to be here to participate in this symposium, and I'm going to try to walk you through briefly why networks are important in trying to achieve the goals that Masanori so nicely outlined. We begin with the notion of reductionism, which serves as the basis, the philosophical and practical basis for research we've all conducted for many decades. It is the basis for the scientific method after Democritus and Descartes. We're in search of ever more refined associations that we believe ultimately define causation. But there are clear limitations that have been increasingly appreciated in this era of omics and big data. And despite statistical attempts at overcoming those limitations, such as weighted GWAS and polygenic risk scores, these do little to solve the problem of complexity in biological systems, largely because they can't address the many interactions that exist at the genetic, genomic, and even phenomic levels that are responsible for disease. I'd like to think about a simple analogy to make this case, and that has to do with how one decides whether or not a machine is functioning properly. Here we have a normally functioning engine with all of its gears and rods. And by comparison, a malfunctioning engine of similar component and composition is found to have, in this case, 3 different gears that are chipped or worn. But we can't tell by looking at those gears whether or not they're responsible for the engine's malfunction or whether they're of no important consequence to function. That very same issue really arises in modern genetics and genomics. We have 4 to 5 million variants in each of us per 3 billion base pairs, the great majority of which are of no functional consequence. But some are, as was illustrated, for example, here in Crohn's disease GWAS study published in 2007 from the Wellcome Trust analysis. Our goal is identifying these functionally important genes with regard to disease. To do so really requires more than a knowledge of a list of the genes and their variants. It really requires a notion of how these genes assemble into a network to govern ascertainable phenotypes, much like the importance of the assembly diagram of this engine. Network medicine provides that kind of assembly guidance. Here is such a network. This is a network, a micro version of a network that governs thrombotic cerebrovascular disease. And as my colleague Wusheng Wang has demonstrated, we can easily illustrate how we can build an assembly diagram using these network principles. These principles have really been established as the basis for network medicine, which explores and really relishes the exploration of complexity in biological systems. And much as Dr. Aikawa pointed out in his talk, this is a truly highly interdisciplinary field that requires skills in computational biology, in statistical physics, in applied mathematics, as much as it does in medicine, or in our case, cardiovascular medicine and biology. We begin with this basic notion that my colleague Laszlo Barabasi and I first posed in a Nature Genetics paper in 2011, that if one looks at the universe of interacting proteins in a cell or an organ, there exists within that universe, as an hypothesis, discrete networks or subnetworks or modules that are tightly linked to disease phenotypes. And that hypothesis was proven to be correct in our analysis of 299 diseases, using as a basis for the analysis the protein-protein interaction network, or interactome. As a result, network medicine does define disease without oversimplification and within all of its rich complexity of genomic and environmental determinants. I said I'd come back to this cerebrovascular disease proto-module to illustrate how we generate it. We begin first by identifying a disease phenotype of interest, and then we ascertain network components or seed proteins using a protein-protein interaction network as the platform for this analysis. And we obtain those seed proteins from existing data, either experimental reductionist data or large data sets that are unsupervised in their acquisition. We map the seed proteins to the interactome and then identify connector proteins, shown as gray nodes here, that link the seed proteins. That's really the key issue behind the information that's provided by this network analysis. These connecting proteins provide the missing links among disease-associated proteins that often lead to the definition of driver pathways and potential drug targets that wouldn't have been appreciated by more conventional strategies. So over the past 20 years or so, there have been numerous examples of using these network approaches for the discovery of mediators of, in this case, cardiovascular and other diseases. The citations for each of these is listed on the left, but they range widely from preeclampsia to pulmonary hypertension and perivascular fibrosis, to SARS-CoV-2 and pulmonary hypertension coexistence, the determinants of their coexistence, to a variety of autoimmune diseases and so forth. And the unique perspective that the network analysis provides really does give us insight both into biological mechanism, but also into therapeutic opportunities. So network medicine is a true path to precision medicine, which I'll try to show in this next example. And that is an example that analyzes hypertrophic cardiomyopathy using a network approach. As many of you on the call know, mutations in more than 11 sarcomeric proteins with over 1,400 variants are associated with the phenotype, but the penetrance is highly variable. And what's often not emphasized is that only about 1/3, more recently, probably closer to 40% of patients with HCM as a convergent phenotype have sarcomeric mutations. So that really does lead us to think that one must, given this highly incompletely penetrant phenotype and the lack of clear primary driver genes, we really must use a network approach to understand the driver pathways in each specific patient case. And this work that was performed by Rusheng Wang and Andre Samakin with Brad Marin and me began with taking myocardial tissue from rejected donor hearts, healthy controls that were technically not feasible for implantation, and obtained RNA-seq data, generating a Pearson correlation matrix between each pair of RNAs. We then took myectomy samples from patients undergoing septal reduction surgery with HCM, 18 different specimens, and performed the same RNA-seq analysis, and then explored on the basis of each individual patient's RNA-seq data set, an analysis of the perturbation of that correlation matrix from normal tissue to rank order and generate, by mapping those rank ordered differentially expressed genes of individual importance to the HCM disease module, as we did here, illustrated here, to create individual patient networks for HCM, or reticulotypes, as we call them, after the Latin for network. Here at very high altitude, you can see those networks from 18 patients, and if you look closely, there are significant differences among them. We explored various intermediate phenotypes and their correlation with some of the pathways that are illustrated or emphasized or responsible for the individual patient reticulotypes, and found some interesting hits that weren't previously appreciated, such as JAK2 being an important driver of this phenotype, in this case, this fibrotic phenotype, in about 6 of the patients listed here. So we think that reticulotypes have a place in the overall link between genotype and phenotype in cardiovascular and other subspecialties of medicine. In moving from the conventional patient grouping on the left through to deep phenotyping and creation of individualized networks, we can then begin to be much more precise about specific therapeutic targets and opportunities in subsets of patients. So network-based precision medicine really gives the information one needs at the molecular level when coupled with precise phenotypic information to begin to identify clusters of patients who warrant approaches that are unique and distinct compared to the overall group of patients with that same disease. Now, why is that important in the modern era? Well, it's important for many reasons, not the least of which is that the overall efficacy of FDA-approved drugs and EMA-approved drugs is quite low. They're approved because they meet the nominal p-value in the registration trial. But yet, as illustrated here from data that were provided by Harold Schmidt to me, you can see that there were, in each case, there are very few patients who are definitively benefited by drugs that range from Advair for asthma to Cymbalta for depression to Remicade for Crohn's disease. And that's precisely because in all of these diseases, we believe that one size fits all. It's commercially simpler to develop drugs on that basis, but it also, in the long run, costs the healthcare system far more because of all of the truly untreated patients who are given a drug of limited value. So network medicine does offer an opportunity to improve upon that weakness and generate, through the steps that I've tried to illustrate for you here, a strategy for isolating and identifying unique drug targets by not necessarily individual patients with a disease, but subgroups of patients with a disease that would respond in a much more robust way to specific therapies that touch on those targets.
Thank you. Thank you very much, Joe, for that beautiful exposition of network medicine and its application. Questions coming through already for Joe's presentation. Among any, please do keep your questions coming on in. We'll try and get to as many as we can in the panel discussion coming up shortly. Meanwhile, our next speaker is Dr. Chiara Giannarelli, Associate Professor at New York University Langone Health. Chiara's research bridges systems pharmacology, systems immunology, and molecular biology to uncover mechanisms driving cardiovascular diseases, including the use of pioneering single-cell proteomic and transcriptomic analyses of human vascular tissues. Chiara, welcome to Frontiers in Science. The floor is yours.
Thank you, Dr. Baker, for the very nice introduction. Single-cell methods are very important to decipher the immune system in cardiovascular disease and other conditions. Immune cells are very remarkably heterogeneous in their function, molecular profile. They vary across tissue, developmental stages, and also environmental conditions. And leveraging single-cell analysis technique like single-cell RNA sequencing enable us to unravel the diversity at unprecedented resolution, shedding light on cellular subtypes, their dynamic interactions, and also their role in health and disease. Why using single-cell sequencing is important, or single-cell analysis in general? It's because these technologies can provide a detailed transcriptional or proteomic profile in individual cells that enable the identification of unique population or phenotypes within an organ or tissue. And as you can see here, unlike bulk analysis here at the bottom, which averages gene expression across all cell types, single cell reveals subtle differences in gene expression across different cells, and this helps distinguish rare subtypes and states that are critical for understanding cellular function, development, or pathological processes. And this is important because using the combination of different type of methodologies that we have now available, we can really have in-depth and high dimensional understanding of the cells within tissues. And in particular, these technologies now allow us to use advanced approaches to study human tissues directly in human specimens, understanding the heterogeneous cell compositions and cells that can have important roles in driving the disease and health. And through data integration, I will explain how the identification of molecular targets can help and aid drug discovery. The ability of single-cell RNA sequencing to uncover cellular heterogeneity, it's important because this can allow cellular phenotyping. It can help targeting the cells based on their phenotype or dysfunctional phenotype in the condition of disease. And this can have very important implications for personalized medicine, because you can have information at the patient level and giving insights into cellular heterogeneity and disease states for that particular tissue, for example, tumor biopsies or blood samples that can help identify cell populations that drive the disease at individual patient level. And these insights can drive and guide tailored therapeutic strategies, like targeting specific cell types or pathways that are involved in patients' conditions. As an example, combining site-seq and single-cell RNA sequencing that allows the identification of the proteome site-of, transcriptome and proteome simultaneously, site-seq, and single-cell RNA sequencing, the transcriptome alone, we were able to generate a more comprehensive and multidimensional understanding of cellular phenotype and function. And by combining all these methods, we can gain transcriptional, translational insights into cellular states and function. This is very important because this knowledge can be used for integrating multi-omics approaches that can be revolutionizing into understanding cell biology and normal physiology and disease states that go beyond what individual modality can achieve alone. And for example, we used site-of to study human atherosclerotic vasculature, and we found through this analysis that T cells are very important in advanced stages of the disease, like human atherosclerotic lesions at the in patients that are undergoing surgery. And we were able to find a specific expansion of these cells in the tissue compared to blood. And based on this analysis, we also were able to identify those the heterogeneity of these cells, as you can see in this separate analysis, where using a similar approach that analyzed the blood and T cell immune compartments, we identified several T cell metaclusters, revealing an heterogeneity that goes beyond what was previously appreciated using traditional analysis of these tissues, and identified particular T cells that are expanded, expressing tissue residency markers and activation markers, along other very specific genes and proteins that distinguish them as expanded in atherosclerotic tissue. And going beyond these points, we were able to identify clinical association, noting the expansion of a particular type of CD4 T cells in plaques of patients who had a recent stroke, as opposed to patients that underwent the surgery but didn't have a recent stroke, indicating that these cells may have a role in driving plaque disruption and leading to ischemic complications of the disease. And using this single cell analysis, you can have several tiers approach that, as I mentioned earlier, when they are integrated together, can lead to deeper understanding of cellular biology and understanding how differences in cell function and behavior can lead to disease states. We also used our approaches, single cell analysis, to leverage this knowledge for drug repositioning to treat atherosclerosis. And this is a concept that it's essentially very important, and it was mentioned before, that drug repurposing can fast track the use or the identification of existing drugs for new indications. And what we did, we perform an ex vivo perturbation using patients immune cells and triggered with their own plasma or plasma from healthy donors, and were able to identify transcriptional inflammatory gene signature using transcriptomic analysis, but also using sight of the exact intracellular activation of signaling pathways that underlie their pro-inflammatory states. And using single cell analysis in this heterogeneous sample of different immune cells in circulating blood, we identified that these changes were occurring in monocytes that we heard from Dr. Aikawa are very important for disease states. And using this signature, we then leverage the integrated computational approaches. We leveraged the links database and identified several candidates drug that were then selected based on their developmental stage, prioritizing phase 2A ready drugs. And we then used a screening that was essentially the same site of analysis that we used for discovery, using patient samples, allowing us to identify the drugs that was capable of preventing the pro-inflammatory signature that we identified to begin with at the individual patient level. And this is drug S, as you can see here, that was able to reduce the effect of the pro-inflammatory signature identified. We decided also to move forward because, of course, it's very important to understand that we still have a need for in vivo validation. So we resorted to a rabbit model that is very important in the field because this model has a very has a lipid profile and type of plaque development that resemble human lesions. And we looked at how this drug, saracatinib, that we identified, was able to reduce macrophages plaque content and reducing, in fact, the inflammatory state of these lesions and also plaque size. And then we moved forward into combining this histological analysis with a clinical molecular readout that can be performed in the clinic using 18 FDG PET MRI or CT, where you look at the signaling of the sugar, 18F FDG, in the arterial wall. And you can see here that in these rabbits, there was a significant reduction of inflammation at the plaque site as well, reflecting the histological findings. Thank you.
Thank you, Chiara, very much. Alrighty, we're out to our final speaker today. It's Professor Jean-Luc Balligand, Professor of Medicine at the Catholic University Louvain in Belgium. He's the past president of the Institute of Clinical and Experimental Research at UC Louvain and a practicing physician at the University Clinic Saint-Luc. Jean-Luc is an internationally recognized expert in cardiovascular nitric oxide synthesis, cardiovascular cell signaling, pharmacology, and network medicine. Welcome, Jean-Luc. Please go ahead. Over to you.
Thank you for the kind introduction, Lee, and it's my pleasure to continue on this forum by addressing some aspects of the innovative and translational aspects for the therapeutic research in cardiovascular diseases. So it's interesting to look back in the past and see what the evolution has been for cardiovascular care. What's illustrated here from a publication recently appeared in Circulation is a graph with the number of deaths per 100,000 people on the y-axis over the years, on the x-axis. And it's clear that since the 60s or 70s, there's been a significant decline in the number of deaths, mostly attributable not only to the development of interventional techniques, thrombolysis, but also the use of well-characterized drugs such as beta blockers. Now, this apparently contrasts with the more recent evolution since 2010, where you see an apparently completely flat curve, despite the description of success for some recently developed medications in RCT and important RCT trials, and not only for sacubitril valsartan in paradigm HF, for example, but also the gliflozins, empagliflozin, dapagliflozins in EMPA-REG and other studies, but also PCSK9 antibodies in the Fourier trials, for example, illustrated on this slide. Now, what might be the reason for this apparent decrease in the success of delivery of cardiovascular care? There may be several reasons for that, including the fact that more survivors from acute myocardial infarction actually now suffering from chronic heart failure, which is much more complex and multifactorial disease, but also a lack of understanding of the biology of the residual cardiovascular risk, including the effect of aging, little consideration for sex-specific aspects, and also influence of the socioeconomic context for efficient implementation of preventive and therapeutic measures. Perhaps this might be also due to a relative disinterest of the pharma industry for cardiovascular disease, given the complexity to put on the market new drugs or therapies, which would need to establish additional benefit beyond current guideline-oriented therapies, which, as we saw, are significantly efficient. So admittedly, we may wait a few more years to see whether the wide but slow implementation of guideline-recommended treatment based on these recent RCTs, positive trials, will really translate into additional benefit. Nevertheless, we've seen this slide before, and I want to make the point again that even though some successful drugs have been developed, they may not be as efficient as we think. And because drugs have not only multiple targets, but also, as illustrated for rosuvastatin, for example, on the lower part of this slide, for every patient that the drug efficiently helps, there are 20 or 19 others that are not efficiently helped. And so this means that one drug fits all, as illustrated amply from the previous talks, is not acceptable anymore. So what would be the potential remedy for that? Well, we've heard the qualities of personalized and targeted treatment that must be based on a shift both in disease definition and also strategies for therapies development that would leverage both on network biology and AI-aided tools. But also repurposed drugs which would have shorter development times and delivered to specific patient groups would both reduce the financial risk for the industrial producers and the costs of healthcare coverage, thereby increasing accessibility to most. This should help to inflect the curve of cardiovascular mortality downwards in the future, hopefully. But how do we implement this? Well, we've had many illustrations before in this webinar. And again, what this schematic illustrates is the sequential steps starting from the combination of different databases, both from biological samples studied by multi-omics, but also electronic health record that then are built to combined to build a large multi-omic database that is then used for bioinformatic analysis to identify new targets and then guide future drug development, identify hit compounds, and lead to the development of new small molecules. But note that beyond new target validation, the new drug developments is also sped up and improved from AI-assisted tools that optimize structure-activity relationship, physicochemical characteristics such as solubility, but also toxicity and potential side effects. So therefore, this altogether will increase the hit-to-lead transition and probability of successful new drug medication development. But nevertheless, drugs have many targets. Actually, a previous study has identified 5 to 1,000 different targets that have been assigned to individual drugs. And in average, more than 30 targets per drug have been identified among the almost 500 FDA-approved drugs. And so this probably explains the frequent side effects because of off-target effects, but also can be used as an opportunity for drug repurposing, as has been illustrated again in the previous talks. But to really speed up this process and also make it more accessible to most for widespread applications, there needs to be the access to tools that would popularize this approach. And an example of publicly available pipelines for such developments is the so-called NetRex pipeline, the principle of which was described in a recent paper in Nature Communication. And key elements for this pipeline is a very large assembly of databases, a knowledge base, including UniProt, NCB, iGene, and other databases, combined with an app for the identification of disease modules from heterogeneous networks, and then using algorithms such as Diamond and others, including tools for statistical validation and identification of a ranked list of drug candidates that either directly bind to the identified target proteins or by proximity on neighboring proteins. Nevertheless, even though the pipeline has been successfully used in some paradigmatic examples, the output still needs experimental validation. But notably, this publicly available tool is upscalable and can be optimized by integrating additional databases, including from tissue or cell-level gene expression data, such as been illustrated in the previous talk. So all this hopefully would lead to more targeted, more personalized drug treatment. But nevertheless, small molecular drugs still have their limitations and may inadequately target nodes that have been identified by network-based approaches. So we need to go beyond small molecules and use additional strategies for targeted delivery, especially if we want to hit target proteins or nodes that are in compartments in tissues or cells that are difficult to access with most small molecular drugs. And so, some new technologies may in part overcome these obstacles by leveraging molecular engineering at the gene, RNA, or protein level. And what this slide illustrates is a number of these new strategies for improved deliveries at discrete target sites. Antibodies have been used for many years, obviously, and have their advantage for very high antigenic specificity or antigen-antibody recognition, and have been used successfully to treat diseases such as familial hypercholesterolemia with anti-PCSK9 antibodies, evolocumab or alirocumab. But targeting proteins can be done with other techniques such as PROTAC, which are bifunctional molecules that combine both a head that targets the specific protein to destroy and a linker, and on the other end, a small part that recognizes an E3 ubiquitin ligase, thereby addressing the targeted protein to ubiquitin proteasome degradation. This is very powerful, but not easily implementable for proteins in all cellular or subcellular localization. RNA therapeutics have had a very big boost after the development of mRNA vaccines for COVID-19, leveraging on a very well established technology for RNA therapeutics, and now siRNA or double-stranded RNA, as well as antisense oligos, have been successfully developed to target several cardiovascular diseases, such as inclisiran, for example, to target again PCSK9 in hypercholesterolemia, or pelacarsen, for example, for an antisense oligo targeting and used for the treatment of the cardiomyopathy associated with amyloidosis. Non-coding RNA include also microRNA, long non-coding RNA. These have had perhaps less well-developed translational applications, but probably because of more complex biology, less well understood for some of them. But long non-coding RNA are more and more listed as potential biomarkers associated to specific cardiovascular diseases. It's worth mentioning CRISPR gene editing with first clinical application that was recently published in the New England Journal of Medicine for thalassemia, by just changing a nucleotide in a inhibitory sequence in the promoter of the hemoglobin F gene, allowing then the compensatory upregulation of the production of this fetal hemoglobin that compensates for thalassemia. And finally, aptamers, so short hairpin RNA oligonucleotides that can specifically target proteins, including its subcellular compartments, such as metomers, targeting directly mitochondria, that can be bound with a number of different cargos to really direct a specific therapeutic agent to an intracellular or extracellular targeted compartment. All of these technologies also benefit from the development of other delivery strategies, including biocompatible nanoparticles with GalNAc that target them to the liver, and also calcium phosphate nanoparticles that can be inhaled, for example. There are examples of that for the treatment of pulmonary hypertension, and as well as, again, leveraging on the specificity of antigen antibody recognition with nanobody directed delivery. So the collaboration between different partners is absolutely necessary for precision targeting. And this needs to rely on the selection of particular patients, and that's where clinicians are very useful, and combining different characteristics of phenotyping from sensor signal, medical images, but also multi-omic results from the analysis of biological samples can be combined using AI tools and AI algorithms to help cluster patients eligible for a specific treatment, as has been illustrated in the previous talks. A caveat, though, for this approach is the need to have high-quality source databases, not only in terms of multi-omics, but also in terms of the clinical phenotypes collected from the patients. And all these databases need to be curated to have optimal and meaningful results. And finally, that's where the collaboration of multiple stakeholders ultimately will allow to combine, the elements that I just mentioned, but also political prioritization, investment funds, academic strategic decisions, all guided by needs that are expressed by patient associations to orient the research towards this new approach, and also assemble international curated databases that should then be exploited by new bioinformatic tools in order to hopefully deliver optimized care accessible to all. And I'll finish this and we'll be happy to answer further questions on any aspect of what I covered.
Thank you, Jean-Luc, very much indeed. And indeed, thanks to all our speakers today. We're now going to move into the panel discussion without any further delay. Please keep any questions coming in. We have many already, and we'll answer as many as we can. First of all, I'd like to introduce two guests additionally who are going to contribute to the panel. First of all, Professor Hugo Ten Cate is Professor of Internal Medicine at the School for Cardiovascular Diseases at Maastricht University. His research has significantly advanced our understanding of coagulation's role in cardiovascular diseases. He was a peer reviewer of the Frontiers in Science lead article that we've just been discussing, and he contributed a viewpoint to the article hub. Secondly, and lastly, Professor Amam Mbakwem from University of Lagos in Nigeria. She's Professor of Medicine there and a consultant cardiologist at the teaching hospital. She's a past president of the Nigerian Cardiac Society, and here is also on behalf of the World Heart Federation. She's the vice president. And she also contributed the policy outlook to the article hub that we've been discussing. So a very warm welcome to you both. I can start with you. We have, before we dive back into the methodologies and the technologies that we've been discussing, just to get the bigger picture, in particular from your perspective, representing the World Heart Federation, in your policy outlook, you refer to the complementarity between precision approaches and population level approaches, of course, which have been the mainstay so far. Could you just elaborate on this a little bit for us? Where do the precision technologies that we are moving toward fit with respect to population level strategies to address the burden of cardiovascular disease globally?
Thank you, Lee, for that question and for inviting me to this meeting. At the WHF, we believe in cardiovascular health equity for everyone. And then we think there should be equity in prevention and treatment, equity in implementation and knowledge, and then also innovation and technology. And if you look at the WHF 2030 policy document, it talks about leveraging technology, to improve cardiovascular care. So I think it's a low-hanging fruit, for us to leapfrog what is happening, what's been developed, what's been seen, to improve care in low and middle income countries or poor resourced areas. But, we need to get in data. We have been talking about precision medicine. We know that epigenetics, genomics, and everything is very different. I mean, we are a practice in Africa. I mean, it's totally different. So for us to do that, I think we need to get data from Africa that needs to be included in all this machine learning and AI algorithms for it to be, really useful. Because if not, I think it's going to further widen the gap between the global north and the global south. Thank you.
Thank you very much, Amam. I think we'll come back further on to implementation challenges later. The lead article discussing a sort of a policy framework and global health leadership. So thank you for that for now. Hugo, as I mentioned, your particular expertise is in thrombotics, particularly precision. Well, could you give us the perspective on how precision medicine is necessary and increasingly being applied in antithrombotic medicine? Of course, there's been some personalization over time with respect to coagulation status, but what's the current situation, and more importantly, perhaps, what's the future in the coming years for precision therapy in antithrombotics?
Well, thank you. I think we're at an important point in time because the past decades have shown significant developments in antithrombotic therapy, and the array of medication has expanded dramatically, I would say. So we have much more choice. And we think as physicians and scientists that we know how it works, that the blood coagulation, that we can manage it. But recently there were 2 large trials with more selective anticoagulants targeting one specific protein in the coagulation cascade, factor XIa, in patients with atrial fibrillation and most recently in acute coronary syndrome. And these trials turned out negative. Negative in the sense that either futile or inferior as compared to standard treatment. Now, without getting into any details, I think this tells us an important story, or a couple of stories. And the first lesson that we should derive is that apparently we don't understand how it works. We don't understand if we are doing a trial with 16,000 patients. That was the most recent trial. We intended to include 16,000 patients. And we consider them all as one entity, acute coronary syndrome. And we think that it's all the same, and that we can all target it with one specific, very highly specific drug. And that does not turn to be true. So we need to better understand our patients, the individual patients. We better need to understand better the disease process and the dynamics of disease. And we need to understand when specific target proteins like factor XIa or glycoprotein 6 at platelets are expressed and active in these patients. So here is the point. I think there's definite need for precision medicine to develop. So therefore, I was very happy with this article and this initiative to give us the outlines, the avenues by which we can proceed, combining unsupervised or unbiased techniques with more biased techniques, as we have been doing over the past decades, because the combination may be most relevant and informative in the end.
Thank you, Hugo. I think that brings us nicely, looping us back into the methodologies. Masanori, back to you. Your approach, and as you describe in the lead article, talks about the different scales of heterogeneity in cardiovascular diseases, be they genomic, cellular, and clinical, and the sort of interrelationship between these layers and all being relevant. Unpack for us, where are the methods of precision medicine best equipped to gain understandings here that can lead to improvements in patient care and can inform research and innovation between those levels?
Yeah, thank you, Lee, for such a wonderful question. At present, I think precision medicine may be best equipped to address molecular and cellular heterogeneity. For example, at the cellular level, many studies uncovered how different cell types within arteries contribute to disease in different ways. So these insights, help us to identify more cell type specific therapies, right? And also in the case of the heterogeneity within the same, cell types, like macrophages, we may identify biomarkers that represent the disease driver subpopulation. And some plasma proteomics platforms have relatively affordable small panels nowadays. And also, measurement could potentially be done with a benchtop instrument, relatively quickly, and in the clinic. So therefore, translation of the biology of heterogeneity in patient stratification at cellular level may become a reality in the near future. And also one of the best examples of translating genetic discoveries into precision medicine might be the use of TET2 mutations as a marker of elevated cardiovascular risk. TET2 mutation clonal hematopoiesis, not only increases the risk of myeloid neoplasms, but also drives systemic inflammation largely through enhanced IL-1 beta signaling, which has been linked to atherosclerotic cardiovascular diseases. Notably, and very interestingly, the CANTOS trial showed that IL-1 beta blockade with canakinumab reduced cardiovascular events to a greater extent in individuals carrying TET2 mutations. highlighting the potential for genotype-guided anti-inflammatory therapy. So similar to plasma proteomics, next-generation sequencing panels, including small focus genes, such as those containing TET2, are now commercially available, actually being used in the, myeloid neoplasm space. So therefore, this approach may become clinical reality in the near future in cardiovascular disease too. However, I think dealing with the clinical heterogeneity remains somewhat challenging. Based on our own experience, one challenge with electronic health record is that the data are generally noisy. This makes it a little bit difficult for us to identify subpopulations only based on EHR data. And also another challenge might be EHR data of hospital setting were not designed for research purposes. So therefore, doing proper clustering analysis just based on EHR, require formal cohort study and with a well-defined set of variables to be collected at the pre-scheduled times and a good number of patients. And also in the case of vascular events, probably important parameter is a good follow-up time to capture relevant outcomes. But overall, I think we need to implement all the information from all three scales areas to accomplish precision medicine for cardiovascular disease.
Thank you, Masanori. Maybe it brings us to one of our audience questions, actually. I'm afraid it's an anonymous question. Please do drop your name and we can give you credit. Joe, if I could come to you. The question we've had from one attendee: network medicine approaches can identify key disease mediators, but how can we move beyond correlation to establish true causality? And perhaps we could build into that. I don't know whether you could also maybe comment on, speaking to Masanori's point, the difficulty in multimodal data from different data sets and aspects such as the increasing or the applications of AI machine learning. What advances in machine learning are helping to understand these complicated, noisy, multimodal data sets to try to reach some sort of causality rather than correlation? Please, Joe.
Oh, thank you, Lee, and I thank the audience participant for those interesting questions and issues. I'll begin by affirming a point that Masanori made earlier as well, that any inference that we derive from an analysis of a complex system always must be followed up by actual experimentation in a more conventional manner, either in a cell-based system or in an animal model. That gives us the kind of strength of association we need to draw causation into the picture. And if we're going to develop a therapeutic strategy based on that knowledge, we really do need to know whether or not this mediator or these mediators are responsible for the phenotype. So that's the first point. Second is that in contrast to the way we typically would go about analyzing multi-omic data sets or even single-omic data sets now, where we look for one-off associations after making the appropriate statistical corrections. In those settings, without a time dependence or pseudo-time, we really can't say very much at all about causation. And the landscape for exploring those potential relationships to causation is much broader than will be the case for a network medicine-based mediator. And the reason I say that is in the context of the network architecture within which this mediator sits, we also can appreciate or understand the pathways that it participates in and explore based on rational biological curation, whether or not those pathways make sense for the phenotype. Now that brings us to the second question or set of questions you raised specifically about how AI might help us here. And what AI is really good at is identifying patterns in complex data clouds that we can't perceive either visually or with more conventional statistical methods, even those that are a little further advanced with multivariable approaches. And the beauty of that is that one can now use AI informs network science, and network science informs AI. There's a mutuality there that enhances the output. So from the perspective of causation, an AI analysis of a complex network within which there's a perturbation that you feel is associated with a phenotype can much more likely tease out the underlying mechanism that might allow for the translation of that association to causation. Similarly, if an unsupervised AI or machine learning based approach is used to look at a large complex data set or set of data sets, the time consumed, the misdirection that exists during the course of that analysis, and the ultimate production of information that may or may not be useful is much more limited, if you will, than if that AI analysis is guided by a network infrastructure. So these are tightly linked now. In fact, there was a symposium some of us participated in last year that was supported in Milan, that explored artificial intelligence and network medicine as a path to precision medicine. This was the Menarini Foundation provided the support we needed to create this symposium, which helped really begin to address those very specific issues that you raised.
Thank you. Thank you, Joe. Chiara, again, going to the audience questions, we've had a question specifically related, perhaps again, bridging the translational aspects that we've spoke about both with Masanori and Joe just before. How do we quantitatively translate preclinical findings in plaque reductions, for example, in the rabbit model shown, into predictions as to whether they will lead to meaningful clinical outcome changes in a cardiovascular trial? So again, challenges and opportunities in translating, particularly in your single cell work, toward clinical implications.
Yes, this is a very important question, and it's something that as a researcher we think all the time. What we do know is that we need this type of research, basic research, to understand the effect in a system models, as I explained. The reason in this study in rabbits, we used imaging, it's exactly this, because we decided to use not just tools that are available in preclinical research, but really a clinical grade imaging modality, particularly 18-FDG PET MRI in this case, but it will be the equivalent of a PET CT that is routinely used in the clinic. And the reason being that we do know from a multitude of different studies in patients that by reducing plaque size, we can reduce events, and by reducing plaque inflammation that can be detected by the uptake of FDG at the vascular site, there is a ability of identifying plaques that are inflamed at a high risk of rupture. And by detecting this plaque, we can stratify patients based on their risk for rupture and cardiovascular events. And we also do know that by reducing plaque inflammation, this risk can be reduced. So putting all these pieces together, we wanted to use a translational imaging modality in our preclinical study that directly translates into humans. And I would like to add that this imaging modality are being used in human clinical studies as a surrogate endpoints for understanding and investigating the effect of drugs in reducing cardiovascular risk in patients, often as a sub-study or a study performed before moving into large endpoint phase 3 clinical studies. So it's a very important message to keep in mind that as we perform this type of investigations, they need to have implications for the real world. This is also a clinical imaging data that are available as they are performed routinely in the clinic.
Thank you, Chiara. Speaking of clinical implications, the lead article discusses the challenges and obstacles to drug development, and specifically devotes attention to the promise, the prospects and successes to date of RNA targeting therapies. Jean-Luc, if I can come to you, can you briefly touch on what are you mentioned in your presentation some of the applications so far of RNA therapies in cardiovascular disease, lipid metabolisms in particular. What's the future? Where are the new opportunities in RNA therapies? What's the state of play, and what can we expect in the coming years?
Well, it's already very well developed. As I mentioned, it also builds on a biology of RNA that's been studied, since the 80s. So, and certainly the recent development of the mRNA vaccines for COVID-19 has given a big boost to the application of these technologies to not only vaccines against other viral diseases, but also using RNA therapeutics for many other diseases, including cardiovascular diseases. The advantage is that compared to a small molecule drug, here the RNA technology directly targets the translation, or thereby the production of a specific protein. A big advantage is that it's very flexible. Once the sequence of the target RNA is known, it's relatively straightforward to produce the oligos that would target it. The challenge is and will always be the efficient targeting, not only to a specific tissue, but to a specific subcellular compartment. But there have been recent development using bioconjugation, that is conjugating the double-stranded RNA, for example, into a lipid nanoparticle to a conjugate, be it an aptamer or another driving molecule that would directly lead the cargo to a specific cell, thereby enhancing the specificity of the treatment. There are examples of that already, even though few are in clinical development so far. But there are distinctive advantage that makes us hopeful that these RNA-based therapeutics will certainly be one of the main direction for future translation of all the science that we've talked about. Also including the fact that in terms of pharmacokinetics, the RNA therapeutics seem to be less complex than the pharmacokinetics usually tackled for the development of small molecules, especially because RNA therapeutics are not metabolized by cytochrome P450 isozymes, for example, or not bound to transporter proteins. So this makes the steps from the development of a specific oligo to its development for clinical application much more straightforward, even though there are still caveats, obviously because of potential toxicity, that these RNAs or oligonucleotides are still amenable to degradation by nucleases, and some of the fragments may themselves have some off-target effects. So all this needs to be carefully evaluated, obviously, but the simple development, I mean, the fact that many of these RNA therapeutics are already now not only in the clinical arena, but also under development in many cardiovascular diseases. I mentioned some of them. Inclisiran is well established, belacarsen, but also aptamers, for example. There is one in phase 2 development for dilated cardiomyopathy. I haven't mentioned also the use of microRNA, which is another kind of RNA-based therapeutic. Even though, again, as I mentioned in my talk, the biology is much more complex because the microRNA may have
Thank you, Jean-Luc. Last few minutes, I think if we could focus a little bit on broadening back out implementation. How is the transformation to precision cardiovascular medicine? How do you see it happening? Hugo, in your viewpoint, you alluded to the fact that precision medicine was sort of born, if you like, in oncology. Genomic genotyping of tumors led to a revolution in that sector, in that field of care. How do we see the transformation in cardiovascular medicine? Is there the same incentives? What kinds of what does your field need to be telling policymakers about the importance of driving this transformation? Hugo, could I start with you on that? You made that allusion in your viewpoint. From your perspective, how do you see this being incentivized, if you like, by policymakers?
Well, I think what oncologists have appreciated over time, and that's a lesson that they learned from the uniqueness of the tumor, is also to appreciate the uniqueness of the host, of the patient, in fact, and also learning how to address in a more personal manner the uniqueness of a disease and disease pattern, and also to limit toxicity. And I think that's the main lesson that we start learning, and those points were also addressed by the previous speakers, by numbers needed to treat and to harm. Is that we need to identify individual characteristics in order to better address efficacy, but also toxicity, and to limit toxicity. That becomes really an important issue, certainly also in the field of antithrombotic agents. So I think that's a key lesson that we learned from our oncology colleagues to focus.
Absolutely, thank you, Hugo. Amam, can I come back to you? Of course, you're vice president of the World Heart Federation with a global perspective of global health. You're also working in the University Hospital Lagos, Nigeria. Can you give us your sense of the policies and the sort of collaborations that are necessary to drive precision medicine research and innovation in Africa and other low middle income countries? How do we ensure that these innovations, technologies benefit the global south as well as the global north?
So thank you. I think the starting point will be to get quality data from Africa and the low income countries like Asia. For instance, in Africa, here we're talking about the proteomics. I was quite intrigued by the macrophage policy, but we know that in some of these low-income areas, infection is still a problem. And so there's inflammation from infection apart from the cardiovascular disease we're talking about. And we've seen it in the phenotypes because cardiovascular diseases, especially in Africa, happen like a decade or two earlier than in the north. And it's not just the risk factors that are not controlled. There are so many other things going on. So we need quality data from these areas to build into the precision medicine. And, it's quite interesting because I think the precision medicine will work for us because a lot of the medications, that have worked so much in the global north, we haven't had such successes, in the south. So making sure we have data that will go into the deep learning and contributing into the omics, all the omics that we're looking at. I think we need to get to that point for everybody to benefit, from the good points that come out of precision medicine and for better outcome in cardiovascular health. Thank you.
Thank you, Amam. We are almost up to time, believe it or not. It's gone very quickly. I'm going to ask each of you to give me your last thoughts. Again, perhaps we could stick to this. If you could offer one message to the broad audience we have across science and policy in society, what's your vision for the expansion of precision cardiovascular medicine in the coming, let's say, 5 years or so, to really drive research and innovation? Perhaps if I could start with you, Jean-Luc.
Well, I think we've heard the power of using network medicine-based strategies in preclinical research, mostly in academic environment. I think that to translate all this new knowledge into new therapeutics, we need to, as highlighted by Masanori in this talk, we need to have collaboration between different stakeholders, not only with public health agencies to define priorities for new targets, but also with the industry that certainly can help with upscaling the development of these new biological therapeutics. And there are several tools to do that at the governmental or institutional level. Certainly in the European Commission, there are now institutional tools that would provide money to consortia that would necessarily associate academic labs to specific industries based on a common agenda towards the same objective. I think that we will need to go through that if we want to really come to meaningful outcomes accessible to most people suffering from cardiovascular disease. Yeah.
Thank you, Jean-Luc. Last word to Chiara, please, from Chiara.
Yes, we heard a lot about precision treatments and discovery on new drugs or existing drugs. I will also like to mention that precision medicine is also about understanding prevention better. And this is something that we didn't have the opportunity to expand upon, but I believe that this is an opportunity for everyone to collect data and trying to understand how through policies and through changing habits and healthy habits and exposure to different type of pollutants, we can reduce risk, cardiovascular burden and cardiovascular risk. So I would like to advocate for thinking about prevention measurements as well.
Thank you very much, Chiara. Great point. Hugo, please, if you would.
Final words, then I would like to support Jean-Luc's words and say that the collaboration is key, especially between clinicians, scientists, and people within the industry, even the investors, I think, because setting up these trials is hugely expensive. And we cannot afford to continue embarking on trials that have a substantial risk of failure. I mean, we should really invest with all the stakeholders in development, joint development of precision medicine to get the best chances for profitable outcomes for all parties.
Thank you. And Joe, please, your last thoughts.
Yes, I would add to these comments the fact that we really need to redefine the paradigm for drug development. Industry needs to be involved in that. And I would say that one thing that network analysis provides is the following: that we all have orphan diseases. No two people with heart failure have the same phenotype nor the same underlying genomic landscape. And that while it's not commercially viable to find precise therapies on a personalized basis, it would be if one were to collect sufficient numbers of patients with key drivers of the phenotype. That's quite different from what the pharmaceutical industry has been used to doing. It does know how to develop drugs for orphan diseases, so it has that infrastructure, if you will. And we need to make sure that there's sufficient incentive for them to follow along or be willing to follow along without breaking the bank or making it impossible for folks, especially in underdeveloped parts of the world, not to receive the precise treatments they need as well.
Speaking, last word from the World Health Federation. Amam, if you would, before we close up with Masanori. Amam, please.
So thank you. So for me, advocacy is still key. We shouldn't forget the basic things that we know, the risk factors that lead to cardiovascular disease. I know precision medicine is the way to go, but even as we're saying, because precision medicine, because one size does not fit everyone, one size will not fit every country. So that each country in developing their cardiovascular plan, should work out the precision medicine aspect that fits their purpose, and their objectives in that country. But advocacy to get the policymakers to understand what we're talking about, and not just in the laboratories and in the journals. Thank you.
Thank you, Amam. And lastly, Masanori, you brought this editorial project to Frontiers in Science. Thank you again. Last word to you.
Okay. Our article in the beginning emphasized the challenges, opportunities. So this is my personal philosophy too. And let's on the bright side and to work together to create the effective synergy between emerging technologies and new paradigms to accelerate the translation of discoveries into precision medicine. Thank you.
Thank you very much, everybody. So we reached the end of the session today. I'd like to thank Professor Masanori Aikawa, Joe Loscalzo, Chiara Giannarelli, Jean-Luc Balligand, Hugo Ten Cate, and Professor Amam Mbakwem from the World Health Federation for all your insights today. Thank you, of course, for you to listening. You can here is that do please visit the Frontiers in Science website, where you can read that article and all the surrounding material. There's also 2 excellent editorials. I do urge you to read those too. Thank you very much for your attention. Thanks for being with us today. Thanks to all presenters. One last wave, and we look forward to seeing you next time. Bye-bye, everyone.